is the aqueous environment, the water, your blood. This will be able to float around in it. It's the hydrocarbons. If there's ever an example, see these hydrocarbons as they call them, say leucine for example. These are oil in water they call them, aliphatic. They do not dissolve, they do not like water, they call them hydrophobic, they're afraid of water. Some say water loving, water hating. I don't know if I want to go to that extreme. But doesn't like it. Oil and water. That's where you need an emulsifier. People that cook, what is it, mayonnaise and something else, you got to get to make vinegar, I guess, isn't one of them, isn't it? There's oil and another one that they get to make mayonnaise. I don't prefer like it, so I don't care. Anyway, we're back to the polar. This is an aldehyde. So this is what I wanted to show you. Even I make mistakes. We drew that aldehyde like it was an alcohol, right? There's no proton available to play in the game. So what we want to do is just color that in all together, see if that'll work. Okay, there we go. Okay, so an aldehyde on the end. See like that there? If this stopped and it just had a red dot, so this is considered an aldehyde, not. And then what I want to do too, we'll keep this all together. If we're writing polar in red, we're going to write polar in red. So I'm going to show you I make mistakes here too, folks. Keep all the polar in red. We got the alcohols. Now what we can do, I was going to say, see, watch how I do this. We'll do this together here. Keep all the alcohols drawn with the dots. So here's our circle. See, these are recycled markers. That's why they're all beat up. These are my good ones for the whiteboard, and it's no wonder the whiteboard's ones don't work very good, because look what I do to them. Okay, kind of leave that way. So now all our alcohols have that on it, and that really didn't do too good with the green there, did it? That doesn't help much either. And this is how they're strung together. Remember, the carboxyl nitrogen is how we drew these. It's like an arrow coming in. See, there's the nitrogen head, the Schroeder diamond holding the amino acid, carboxylate tail. Now, isn't that cool? I mean, this looks artistic to me. Pick up the microphone here. This looks cool. The way they draw it, I mean, this is how my notes, I showed you this before, the structure. See how they're just lines with the letters and, you know, I mean, it looks boring to me. I understand it from doing it for years on end. But what looks better? That, as you see, we're going to draw these and then we'll be able to compare them. So again, as we come through, we're going to try and get each amino acid here and notice the chemical characteristics of it from the art. So when we look at glutamine here, we don't care too much about the side group, or the backbone, the peptide bonds. Okay, that stuff there, we don't care too much about. What we care is the side group. Look at that baby. It's got the nitrogen alien, and look, the hydrogen's giving like little arms on the end. Then if you drew the lone pairs, because you know when I talk chemistry, I'm the stickler on the electrons. I'm keeping this simple by not doing it on those other ones over there. But look, it's kind of like two aliens on the oxygen, a single on the nitrogen. We're going to do an alien thing for you guys. I know everybody likes the aliens. God will forgive me. Let's see, what do we got here? This is glutamine. Next one we had over here was aspartic acid. This one, it said, was electrically charged. Remember, there's the backbone. Now the side group, oh, it's a carboxylic acid. what I call carboxylate, they'll draw a double bonded, and this is the OH. It's one way to draw a carboxylic acid. So aspartic acid. I'm going to try and draw a plate here with it. I'm no good at doing this. Okay, so we're just going to, this would be easier to do this way, wouldn't it? So there's aspartic acid. There is glutamine. Here is asparginine. Here is glutamine. Here is tyrosine. There's another alcohol. Serine. Here's another alcohol. Threonine. Here's another polar, the sulfur containing. Cysteine and methionine. Over here was something we were trying in the earlier days, just to show the backbone, because see, these are the simplest amino acids here. So glycine, if you recall, forget that line in there, simply has the one proton on the end of it. So here is the backbone. This is a good example to show the backbones. So the C here I wanted with the O, I was writing as CO to distinguish because there's two carbons in there. So simple glycine, here's alanine. So the CO again I was drawing in there because if you remember how the arrows come in, we'll show that later. We're just going to go through the amino acids here again. So here's this one methyl group on the end, alanine. Here's two methyls on the end of a carbon here. That's valine. Then there was leucine. This is the one that's going to be our isomer, if you recall. Leucine has the same number of carbons as isoleucine. So let's put these next to each other and look at them here. So you can at least read it. Okay, I'm going to keep the nitrogen in there so it looks good. So can you compare them? I'm cutting out the nitrogen. Leucine, isoleucine. Same number of carbons, 4. But see how they're different. Now see the beauty of these cards? I mean, I'm trying to keep the N in here to keep this legit. But see the way they go together and compare them. Now ideally what I want to do is I want to cut these out, but then the name would fall off. So I'm not doing that for now. So there is isoleucine. And my original discussion, what I was saying was we're calling this the carbonyl diamond ring. See, it looks like a diamond ring. Cool. See, good thing I write these notes down, huh? So this we're going to call the carbonyl diamond ring. Now, where you got to know your chemistry is because this thing is actually the carboxylic acid drawn like that, but they draw it with the OH on the end of it. Can I get it up there right in front? Right there, like that, right? Well, that OH is what gets hydrolyzed, makes water in the peptide bond, so now what you're left with is just a carbonyl group up there. That other oxygen's gone because that bonds to the nitrogen, remember? When along comes the nitrogen over here. So where'd that O go? There were two O's on the carboxylic acid, right? Well, that one went to make water when this boinked together, when the arrow shot in. See the way we do this?